think, to round off the year.
I like some of the Cliff Richard ones, but they're probably a bit dated now.
Yeah. No, it's always the classics that are good. Rightio, okay. Shall we look at making a start then?
Yes.
Welcome to those of us on Zoom, and those of us that are joining on YouTube, welcome to you also. This is the final meeting of the year. Can you believe it? How 2020 has flown us by, and what a remarkable year it's been. So there's really no more appropriate way to end it off than a talk on viruses and the connotations that viruses bring to this world. There could be nothing better. So I'll hand over to Dr. Alan Stevens, who's going to talk a little bit about himself and then give us an hour spectacular on the world of viruses. So thanks, Alan. Over to you.
Thank you. Well, I'm going to repeat Ben's very warm welcome to you all tonight, whether you are watching live on Zoom or YouTube or later on by catch-up. I hope you're all well and have been enjoying the previous online presentations from the Womban Science Society and Historical Society over the last 9 months. One compensation about being online is that you don't have to look for a parking space to join the meeting. Anyway, a little bit about me. I've recently retired from companion animal veterinary practice after 42 years, working mainly in the UK as a general veterinary practitioner. And during my career, there have been several new or novel diseases which have emerged to affect animals and humans. And even as I'm presenting this talk, there are outbreaks of avian influenza, but more about that later. So my presentation is entitled Going Viral, A Vet's Eye View. And I'm going to cover 3 areas tonight. Firstly, what is a virus? What do we know about them? How do they work? Secondly, we shall take a look at how they affect us. And for this talk, how do they affect some of the animals which I have been involved with as a vet? And in this section, we'll consider what we can do to fight the bad viruses. And lastly, I'm going to look at the possibility that there are some good ones out there. So what are viruses? Are they alive? Living organisms are defined by the ability to move, to reproduce, to produce energy by various cellular biochemical cycles called respiration. And to do that, they need to take in nutrients and be able to remove or excrete waste products, sense their environment. They need to be able to reproduce and grow. But viruses don't have all these characteristics of being a living organism, but they do reproduce and adapt or mutate through natural selection. Some scientists consider viruses to be non-living or proto-living, somewhere halfway between living and unliving, a bit like the Borg. Fortunately, in most cases, resistance isn't futile and we haven't been assimilated yet, but COVID-19 has been having a very good attempt. So where have viruses come from? There are lots of theories. Did they come from space? As obligate parasites, they need a host cell to replicate. So if there are no host cells around, viruses can't replicate. But can they still exist? Well, it's unlikely, as viruses tend only to survive for short periods of time outside their hosts. One likely theory is that they possibly come from bacteria. Bacteria can pass genetic material called plasmids, which I'm just showing up on the slide here now, between other bacteria. It's called horizontal genetic transfer. And incidentally, this is an important mechanism in transferring antibiotic resistance among bacteria. So this is a 3D model of a virus. You've seen that earlier, swirling around. It's a very good model and it's courtesy of Microsoft, but this will help us to understand the structure of a typical virus. So starting from the inside and working outwards, this is the virus genetic material or the genome. And in this case, it's made of RNA, but it can also be made of DNA. And it's a long molecule, and it's very equivalent to our genetic material inside all of our cells, our chromosomes. It provides the virus with the codes to reproduce protein structures and create new viruses or other messenger-like chemicals such as hormones. And around the nuclear protein is the protein coating called the capsid. This is the green area here, and this protects the nuclear protein. Then around that is the lipid envelope, which is formed from the cell wall, the host cell wall, or the internal host cell membranes. And this adds another layer of protection, especially when living away from host cells. So this virus model here is of flu or AIDS virus or even COVID-19. Not all viruses have a lipid coat, but those that do are vulnerable to alcoholic chemicals, which can disrupt the virus, hence using our hand rubs and so on to protect ourselves from COVID. Externally, there are two types of appendages. One's called hemagglutinins, and that's these red things. And the other ones are neuraminidase, which I can't say after having a drink of water. And these help the virus to evade host cell defenses and also help to invade or penetrate the host cell. These can be actually used to subtype some viruses, especially influenza virus. The most recent avian influenza virus is called H5N8. That doesn't mean they've got 5 of one appendage and 8 of another. It's just the way that they've been subtyped. So what size are viruses? This chart shows the relative size of objects on a logarithmic scale. As one goes from right to left, each division is 10 times smaller than the previous division. Anything larger than 1/10 of a millimeter can be seen with the naked eye. So that's all along here. So all of these objects here can be seen by the naked eye. Anything smaller can be seen with the aid of a light microscope. And included in this group are cells, animal and plant cells, bacteria, and structures within the cells. But the light microscope is limited. It can only go down to a certain level, and then the resolution problems cause the ability to see things more difficult. So we then move on to the electron microscope, and that is able to show up viruses, protein molecules, lipid molecules, and even atoms. Let's have a look at this in a bit more detail. So I've enlarged my scale. So we are now at the bottom end of that scale, and we've got the atoms here. We've got the lipid molecules again, and the protein molecules. And this is a Pandora virus, and it's about the size of a bacteria, and it is visible by the light microscope. So it's not true that you have to have an electron microscope to see some viruses. The biggest ones you can see under the microscope, these affect amoeba, and they're harmless to us, so we don't have to worry about them. Here's our flu virus again. And this is just about under the 10th of the size of a Pandora virus, about 80 to 120 nanometers. Forgive me if you're getting a bit confused by the scaling. The smallest virus is a picornavirus or parvovirus, and they're about 20 to 30 nanometers. Let's look at this in a different way. How many of these things can you fit on a pinhead? 1,680 Pandora viruses, 14,000 to 21,000 flu viruses, 56 to 84,000 picornaviruses, and just for completeness, 6,720,000 atoms. Atoms might vary in weight a lot, but they don't vary much in size. They're about a quarter of a nanometer. And that's why you can fit so many of them on this little pinhead here. There are millions of types of viruses in virtually every ecosystem on Earth. Only about 6,000 have been described in detail, and these are the viruses that have significant effect on us or our animals or our plants. But viruses actually come in all sorts of shapes and sizes. So we have flu and HIV viruses, and they're spherical. Sorry, I'm just trying to get my pointer in the right place. These are the flu viruses. They're spherical. Adenoviruses have a shape, a 20-shaped polyhedron or 20-surfaced polyhedron called an icosahedron or icosahedron, because Greek for 20 is ikosi. So that's why they're named that. We have the tobacco virus, mosaic virus, which is tubular or rod-shaped. This is a pain if you're growing tobacco leaves. It can damage them. And then Ebola viruses are filamentous and quite large. This is a colorized scanning electron micrograph of Ebola viruses. This is 20,000 times magnification, and these greeny yellow things are the Ebola viruses, and some of them in blue are actually budding out from the surface of this green African green monkey kidney cell. So how do viruses work? I'm going to take a brief look at this under 4 headings. I'm going to give a little definition, and then we're going to talk about their reproduction, because their sole aim seems to be to reproduce themselves. So we'll look at this in some detail. And then we're going to have a look at their pathogenic effects. What I mean by that is what disease problems they cause us. And then we're going to have a look at how they get around. So you've all heard of viruses, but they actually, the free living versions are called virions. And viruses or virions can't exist for long in this state. There are some exceptions, but environmental conditions can help or hinder virion survival. And that's why we use all our disinfectants and antiseptics and hand rubs in order to try and make the environment as unfavorable as possible. Their sole aim seems to be to reproduce themselves. Reproduction takes place in cells of susceptible species, which can be almost anything which is made up from cells. So animals, plants, insects, fungi, and other microbes. To get to the cells of choice, viruses have to evade host mechanisms, so physical barriers like our skin, or the immune defenses found in our bloodstream like antibodies and cells, which are programmed to destroy foreign invaders like bacteria and viruses. Once the virions have found their cells of choice, they need ways to get into the host cells. And having penetrated the cells, they need ways to hijack the cell's genetic code. And finally, they need to force the cell to make new virions. DNA viruses and RNA viruses reproduce in different parts of the cell, and I'll discuss that in the next few slides. Let's move my script on. Right, so let's have a look at each stage. So our first stage of reproduction of the virus is attachment. So our virus is now in the bloodstream or in the body, and it's found its cell, and the receptors on the surface of the virus enable it to attach to the cell wall. Here's an example of attachment as shown in this electron microscope image of bacteriophages attached to a bacterial cell. And you can see how they are all sitting on the surface, and we'll have a look at that again in a bit more detail in the next slide or so. So next stage is penetration. Most virions enter the host cell through receptor-mediated endocytosis or membrane fusion. Sorry, these are definitions again. But what endocytosis is that the receptors on the virus fools a cell and causes a cell wall basically to engulf the virus or eat it and take it into the cell's interior. Or the other way is that the lipid coating on the virus fuses with the lipid layers in the cell wall, And once they're fused, the interiors of the once independent structures gain access to each other. But if you don't need to force open the front door, you can go through the keyhole instead. This is what the bacteriophages do. They sit on the cell surface and they then inject their genome into the cell. Bacteriophages are viruses that infect and kill bacteria. Bacteriophages are most commonly found in seawater in vast numbers. So you can find about 250 million of them in 1 milliliter of seawater. And given that the bacterial cell walls are much thinner than plant cell walls due to their smaller size, these viruses have developed the mechanism to inject their genome into the bacterial cell wall across the cell wall. And these blighters can produce 300 new viruses within about 20 minutes. Our next stage is uncoating. And this is a process where the virus undresses itself and releases its genome into the cell. And then you get biosynthesis or replication. And this involves primarily multiplication of the genome. It's a very complicated process with much variation because, as I mentioned before, viruses have different types of virus genome. But the main result is the virus genome being made by the host cell. There can be alternative outcomes, as we'll see. So DNA viruses have to work in the nucleus. So they've not only got to get into the cell, they have to get across the nuclear membrane and into the nucleus. And that's where their genome replication takes place. Whereas RNA viruses work on the outside in an area just around the nucleus, which is normally used by the cell to make proteins. This is co-opted by the virus to make new genomes for the virus. And those genomes then get assembled and create the new viruses all ready and fit to go. And lastly, they get released. And that process can be either by budding, and this is a budding here, and viruses which bud will sometimes get a little bit of the cell wall membrane, and that forms that lipid area that you remember at the beginning of my presentation. But the other way that viruses get out of cells is basically to explode the cells and lyse them. So let's move on to consider some of the ways that viruses cause disease or pathogenic effects on the host. First of all, they can kill the host cell and or the body. When virions are released from the cell by lysis, the cell is killed by bursting its membrane and cell wall if present. And this is a feature of many bacterial and some animal viruses. And if enough cells die, then the whole organism will start to suffer the effects, some of which obviously can be fatal. The other way the virus interferes is with normal cell function. So viruses during their reproduction cycle cause disruption of healthy normal cell function, which is called homeostasis. And this can result in various disease states, which can range from mild to fatal. Some viruses can cause lifelong or chronic infections where the virus continues to replicate in the body despite the host's defense mechanisms. And this is common in hepatitis B and hepatitis C virus infections. People chronically infected are known as carriers, and they serve as reservoirs of infectious virus. In populations with high proportion of carriers, the disease is said to be endemic rather than pandemic. Latency is a situation where the viruses don't reproduce in large numbers, but instead incorporate their genome into the host cell's chromosomes. The viral genome is then known as a provirus, or in the case of those bacteriophages, a prophage. When the host cell divides, the virus genome is also replicated, and the viral genome is mostly silent within the host and evades any defense mounted by the host. And in this way, they may exist relatively harmlessly within an organism, but at some point, the provirus or prophage may give rise to active virus, which may lyse the host cells. And latency is a feature of herpes viruses, including feline herpes virus cat flu, and also several human herpes viruses, including Epstein-Barr virus, which causes glandular fever, and varicella zoster virus, which causes chickenpox and shingles. And I guess most people have been infected with at least one of these types of herpes viruses. Some viruses induce cancers. So you get cervical cancer in women, you get papillomaviruses in animals, and avian lymphoma virus in chickens. You get influenza as well, but that's not quite the same thing. What else can viruses do? Well, they can sometimes stimulate or overstimulate the host immune system. The viruses stimulate a reaction by the host, and in some virus diseases, that reaction becomes like a wildfire and ends up by the host self-destructing in an effort to remove the virus. An example of this is bovine malignant catarrhal fever, but also COVID-19 can cause this in some people. Let's look at how some viruses spread. This is the last section under the heading, How do viruses work? So viruses can spread vertically or by vertical transmission from mother to child. So the AIDS virus, hepatitis B virus, and parvovirus in dogs and cats. The virus transfers from the mother to the developing embryo in the uterus. And we have horizontal or person to person or animal to animal spread. And that happens when body fluids are exchanged, either during sexual activity or when virus infects saliva, urine, or feces. This can contaminate food or water, which is then ingested by the next person or animal. And then there's airborne. You've probably heard of the saying, coughs and sneezes spread diseases. Spread of the virus can occur when aerosols containing viruses are inhaled, and foot and mouth disease in pigs and COVID-19 are classic examples of this. And then another way that horizontal spread can occur, which is through food or bush meat. COVID-19 is believed to have originated in meat products in the meat markets in China, and the meat was probably bats. Ebola virus is another serious condition in humans, which is highly contagious viral hemorrhagic disease that's often fatal in humans. The human outbreaks certainly originated from contact with bush meat that comes from a variety of wild animals, including bats, monkeys, and cane rats and antelope. And in Africa, human infections have been associated with butchering and processing meat from infected animals. And incidentally, it's estimated that every year 1,500 tons of bushmeat is imported into Britain. So that could be a source of viruses. In December 2016, there was an outbreak of avian influenza in New York. A veterinarian working in a cat shelter in New York contracted bird flu, which is an H7N2 virus, from infected cats in the shelter in which he worked. In total, more than 100 cats tested positive for avian influenza across all New York City cat shelters. But this outbreak, the original source of the virus was not determined, but in previous feline cases, infected meat was the source. So whether feeding infected chickens to the cats, we don't know. If you cook meat, it tends to destroy the virus. Then a further way of spread is vectors. And there's two sorts here. There's blood-feeding parasites like mosquitoes, ticks, mites. They can spread virus from one host to another. And then there's a passive method called fomite transmission, Fomite is a passive transfer of virus on clothing, grooming, and other equipment. Virus can be passed on if you touch it or it touches you. And it's a very important mechanism in many virus diseases on man and animals. For example, foot and mouth disease in cattle and sheep. So just for a side line, here are a list of a few veterinary virus diseases, and don't worry, we won't be covering them all this evening, as we'll be here till midnight. This list just illustrates a variety of names that have been given over the year. So we have some named by the appearance of the disease. That first one, SMEDI, which lovely name, isn't it? Stands for stillbirths, mummifications, early embryonic death, and infertility. And that is a virus disease that causes these reproductive disorders in some animals. Then you get blue tongue, and that is because in sheep affected by this virus, which is spread by mosquitoes or midges, they get a blue tongue. And then lastly, there's one called pox, and you probably all know what a pox is if you've had chicken pox. So some are named after the species. So you get canine hepatitis. Some are named after people, like Marek's disease, which is up here. Some are named after places, like African swine fever or Newcastle disease. Where's that gone? There it is, Newcastle disease. And one called West Nile fever. Sometimes further information regarding what effects they have is part of their name. So you talk about respiratory viruses or enteric viruses. So here we have feline and canine enteritis. I'm just going to cover a few of the veterinary diseases that I've come across in my career. Canine Parvovirus is caused by that very smallest of DNA viruses, and infections are often fatal in puppies. The emergence of the parvovirus in the 1970s was one of the most significant in the history of infectious diseases. The disease spread rapidly across the world and thousands of dogs died from the infection. And the virus originated from cats, but a mutation that changed just 2 amino acids in the viral capsid protein allowed it to cross the species barrier to dogs, which, unlike the cats, had no resistance to the disease. It's highly contagious. It's spread from dog to dog by direct or indirect contact with their feces, and vaccines can prevent this infection, but mortality can reach 91% in untreated cases. And canine parvovirus may infect other mammals, including foxes, wolves, cats, and skunks. The virus actually is very tough to get rid of. Bleach is about the only thing that will get rid of it. When it first emerged, it took the veterinary profession by surprise. I was quite a new graduate when I came across my first cases. But as with COVID, rapid action was taken to develop effective ways to treat and control the disease and the cause. Initially, cat vaccines were trialed in dogs. Unfortunately, that worked and helped protect unaffected dogs until a canine vaccine was developed. This is another very serious viral disease of dogs, canine distemper virus. Those of you who have dogs will know that we do vaccinate against these last 2 organisms. Canine distemper is closely related to measles virus. And in fact, you can use measles vaccine to protect dogs, but you can't use dog distemper virus vaccine to protect children, probably just as well. And it's the most important virus disease in dogs. The disease was first described in 1760 by Edward Jenner, who was the pioneer of smallpox vaccination. It's highly contagious, but is well controlled by vaccination. It affects several major parts of the body because the virus attacks the linings of the guts, the airways, and the nervous system. And to diagnose it, one needs to see 4 out of 8 classical clinical signs. It's a bit like COVID, you're looking for classical symptoms. So in dogs with this condition, they may suffer from fever, a persistent cough, a snotty nose, very snotty this one has, severe conjunctivitis, this one actually doesn't look too bad, but you can see the crusting around the eyes, diarrhea, vomiting, because it affects the gut, And if you put your hand on the top of their heads, you sometimes get a little ticking of the muscles, and that's called chorea. And some dogs go on to develop seizures, and if they do that, they're likely to die. And then some dogs who go chronic and recover develop these hard pads. That's why it's also called hard pad. Let's move on to some pussycat disorders now. Affected cats with cat flu show typically heavy cold-like symptoms, lose some of their sense of smell and taste, subsequently losing appetite and sneezing very productively. It isn't usually fatal, but can become worse with bacterial infections on top. Although this is known as feline influenza, this condition actually is not caused by an influenza virus. It's actually caused by two viruses. Feline herpes virus, like our cold sore virus, and chickenpox virus is a herpes virus and is able to hide in the body for variable periods. And I've mentioned this, it's called latency. And from this dormant state, the virus can become reactivated by stress situations. And this reactivation's got a lovely name called recrudescence. So things like a visit to the vets for having their vaccinations, or having treatment for something else, or having a kitty cat holiday in a boarding kennel can trigger the recrudescence, and we get the blame for it. They've caught cat flu when they visited the vets, but it's actually because they've got the virus already on board in their bodies. Kittens are very prone to getting infected if the queen or mother cat hasn't passed on her maternal immunity to them. Even if these kittens are vaccinated, they can be prone to recrudescence, as the virus and the cat's immune system are in lifelong battle for supremacy. Fortunately, most of the time, the cat wins. But if the virus isn't defeated, it's just in retreat until the next stressful episode. The other virus involved is feline calicivirus. And these two viruses, the herpes virus and calicivirus, often work together. This one causes severe ulcers on the tongue. It can also cause lameness in several joints, and you often get a fever. And since 2003, this has mutated further into one that can cause much more severe disease with head swelling and systemic illness with high mortality of 50%. But fortunately, it is rare. I don't think I've ever seen a case of it in this country. You can see how poorly this little cat is. So before anyone had ever heard of COVID-19, cats and dogs both had their own coronavirus infections, but you'll be pleased to know that neither affect humans. So pet owners do not need to worry. So here is feline coronavirus. There are two types of feline coronavirus. There's the enteric coronavirus, and there is the feline infectious peritonitis virus. These are actually the same virus, but the first one has mutated from a fairly harmless diarrhea-causing virus to become this infectious peritonitis virus, which does cause serious and often fatal disease in cats. This virus is able to replicate in the macrophages, and those are the cells which are part of the immune system, whose function is to monitor for foreign invaders and remove them. And it is an immune-mediated disease, i.e. the symptoms arise because of the interplay between the virus and the cat's immune system, which can become overstimulated and then starts to attack not only the virus, but also the host's body. This is a similar situation to what's happening with COVID-19, which is why steroids and other immune suppressing drugs help in the human disease. So often the first thing we see when cats come into the clinic is a depressed cat with jaundice. So you can see here the yellowing around the eyes and also around the ears and the skin in between the ears and the eyes. The jaundice occurs because of liver damage. The virus also causes blood vessels to become inflamed, and that's what's called vasculitis. And then they become leaky. And so sometimes so much fluid leaks, they start getting a swollen tummy. And this is an ultrasound scan of a cat's tummy. So its head is towards that yellow container here, and the tail is that way. And this is the liver, and the black bits here marked FF is free fluid. If we can pop a little needle into that and collect some of the sample, we get this yellow straw-colored viscous fluid, which is very high protein. And we can send that away, and tests can be done to look either for the virus or for antibodies. Our next veterinary virus disease is foot and mouth disease. And I guess everybody's heard of this one as well, but it's not the same as human hand, foot and mouth. This is a different virus altogether. And it's unfortunate that the names are very similar. But foot and mouth in animals affects cloven-footed animals, i.e. the farm animals with split hooves, cattle, sheep, pigs, but also wild stock such as deer. And it presents with lameness and drooling when eating because of the vesicles and blisters affecting typically the feet and also the mouth. There's lesions on the nose, and if you look in their mouth and lips, you'll see them there as well. It's extremely contagious, spreads from animal to animal, but also it's carried on clothing, equipment, and especially via pigs by airborne droplets. One of the things that the government vets always fear is it gets into a pig unit. To control the disease, wide exclusion zones are set up around infected farms. People are prohibited from approaching any countryside in the exclusion zone. The last outbreak in the UK was 2001 in the lead up to a general election, and the movement restrictions had the potential to interfere with the voting. The affected and healthy but susceptible animals are slaughtered so that the infected farms are depopulated. They then are deep cleaned and left empty for prolonged periods of time for the viruses to die off. It is serious because an affected country can't trade in livestock, and those animals that survive are what they call poor doers. In other words, they don't really fatten up very well. Vaccination programs can be used to control the outbreak, but they're not generally used on a long-term basis, and certainly not in this country. Going from the very large to the very small, the honeybee, Apis mellifera, is susceptible to many virus infections, and their numbers have dramatically declined around the world. These bees often suffer from infestations of varroa mites, which are vectors for deformed wing virus. As a result, the virus has become one of the most widely distributed and contagious insect viruses on the planet. The virus causes stunted wings, you just about see them here, and as a result, infected bees are unable to leave the hive and forage for nectar. Symptomatic bees have a severely reduced lifespan of less than 48 hours and are often expelled from the hive by other bees. Bees are crucial to the survival of humans, along with producing honey, because they pollinate plants that contribute to up to 1/3 of the food we eat, and their dramatic decline is a grave concern. One interesting observation has been made, though, that some bees are self-medicating by visiting wood chip piles where certain fungi produce chemicals which can kill the deformed wing virus. How amazing is that? So how do we fight viruses? It's quite complicated, so I'm not going to be covering this in great detail, but our bodies and those of animals have developed sophisticated methods to combat viruses, or at least mitigate the effects viruses have on us. We produce our own antiviral chemicals, one of which is called interferon. Interferons are groups of signaling proteins made and released by host cells in response to the presence of several viruses. In a typical scenario, a virus-infected cell will release interferons, causing nearby cells to heighten their antiviral defenses. Then we have cellular defense. Cell-mediated immunity is an immune response that does not involve antibodies, but rather cell-mediated immunity is the activation of phagocytic white blood cells, like this monocyte, antigen-specific cytotoxic T lymphocytes. The T lymphocytes are killer cells, and the release of various cytokines, which I've just actually mentioned because interferon is one of the cytokines, and they can get released in response to an antigen. And once activated, these cells are programmed to search out for and recognize foreign invaders and destroy them if possible, even if they die in the process. And then we have antibodies. Antibodies are also known as immunoglobulins. They are Y-shaped large proteins, and they're used by the immune system to identify and neutralize foreign objects, such as pathogenic bacteria and viruses. So we have the antigens, which are on the virus or on the bacteria, and then you have receptors on the antibody which match those antigens. And then what happens is when viruses invade, the antibodies latch onto the viruses and lock them all together and prevent them from getting into the cells or doing further damage. And these antigen-antibody complexes are then targeted by the cells, and the cells come along and destroy the whole lot. So having looked at the natural way we can fight viruses, we'll move on to some of our man-made weapons, which are used to fight viruses. Vaccines provide a means to stimulate the body and to produce an immune response against target organisms, which can be viruses or bacteria. The aim of vaccines are to increase herd immunity in such a way that individuals neither develop symptoms of the disease nor become asymptomatic carriers of the virus. There are costs involved in making and administering the vaccines, whether for humans or for animals. So generally they are only adopted for more serious diseases, which, if not controlled, cause their own economic consequences. For viruses, the vaccines can be made from the outer receptor bits of the virus or from the whole live virus, which has to be attenuated or weakened so they can't actually cause disease you're trying to protect against. Precautions and testing has to be carried out to make sure that these modified live viruses can't revert to type. Antiviral treatments, compared to the vast numbers of drugs that we have developed to fight bacteria, historically there were virtually no medicines that would kill viruses, and this has changed in the last 40 years. So we get immune assistance. Some treatments aim to augment and assist the body to neutralize or destroy the invading virus. So we've already seen that the body can produce interferons. This can be manufactured. Virbac make one for cats and dogs. And that can be, if it's given early in the course of virus infection, it can be helpful to combat some of the more serious viruses. And it's also been used to help treat some chronic and intractable viral infections. But in my experience, it's only partially effective and it's very expensive to buy. And another tactic is to give antibodies, which have been derived or from a recovered or vaccinated animal or human, or stimulated by giving vaccines to people or animals and then taking their antibodies from their bloodstream and concentrating it. That's called antiserum. move my slide on. Bear with, there we go. Right, the other way of treating infections or caused by viruses is immune suppression. So some treatments modify the way the immune system works. When working properly, the immune system can be helped and stimulated. But as we saw earlier, some of the immune system becomes dangerously overactive and needs to be suppressed. And you've probably all heard about this in terms of the miracle cure for COVID infection, and that's dexamethasone. It's been used for absolutely donkey's years. It was around when I first graduated, and it's used either by injection or tablets. Other corticosteroids and some chemotherapy drugs used for cancer or rheumatoid arthritis treatments can also be used to suppress the immune system if it's overactive. And then we come to antiviral drugs. And these remdesivir and other drugs have been developed from AIDS and HIV virus research. They have a direct effect on the virus, and they interfere with the processes required to produce their genome, and they halt their replication and their pathogenic effects. So, moving on, what did viruses ever do for us? Only about 6,000 have been described in detail. These are viruses that have a significant effect on us, our animals or plants, because the viruses have led to pandemics and death or economic issues. So their reputation is pretty bad, to say the least. But there are actually millions of types of viruses in virtually every ecosystem on Earth. And are there any good ones out there? So let's have a look at some of the helpful ones. This is the last part of our going viral journey. So let's see if there are any good ones out there. Not all viruses are harmful. Viruses are very selective about which type of cells they infect. So, for example, plant cells don't cause disease in humans, and some viruses may provide benefits to their hosts. We just don't know. Some viruses think that if all viruses magically disappeared one day, which some people might think was a good idea, it might end life as we know it. So viruses can help maintain ecosystems by making sure that individual populations, such as insects, don't outcompete other populations, causing a huge crash in biodiversity. Viruses kill about 20% of microbes in the oceans daily, releasing nutrients from the dead cells, which help feed other microbes. But these killed microbes also sink down to the seabed, locking up the carbon they contain, and thus reducing carbon dioxide levels on Earth. You've probably heard of good bacteria, which help to keep your guts healthy. Viruses may help to regulate bacteria in your guts and train the immune system to fight some infections. Some latent viruses might sometimes be beneficial, as the presence of the virus can increase immunity against bacterial pathogens, such as Yersinia pestis, which, if you didn't know, causes the plague. Then we've looked at these already, the bacteriophages. And the Russians have been finding these in sewage outlets and have been using these to kill off antibiotic-resistant bacterial infections. This is one example of viruses being used as tools in medicine. There should, in theory, be a bacteriophage capable of killing every single bacterium on Earth. If we can match the right bacteriophage to an antibiotic-resistant bacterium, we could treat untreatable bacterial infections. Viruses can be used to deliver correct copies of defective genes into cells during gene therapy. And research even suggests that viruses that specifically kill tumor cells might help us to fight cancer. Controlling insect pests. Baculoviruses, lovely name that, isn't it, are among the best studied of the invertebrate viruses. They infect and kill several species of agricultural pests, and as natural insecticides, they have been used to control insect populations in Brazil and Paraguay, such as this velvet bean caterpillar, which is a pest on soya beans. Viruses also can change the behavior of their insect hosts to their own advantage. A baculovirus of the gypsy moth, and here's some here, make their caterpillars climb to the tops of trees where they die. And in doing so, they release a shower of millions of progeny viruses that go on to infect more caterpillars. So viruses are an attractive alternative to chemical pesticides because they are safe to other livestock and wildlife and leave no residues. So, in conclusion, we've probably only discovered a tiny fraction of the viruses on our planet, with most research focusing on disease. But the more we learn about viruses, the more we understand their importance in the life of the planet. So thank you for watching. If you have any questions, I'll try and answer them. And if you want any further information about the WSS and WS and HS, should I say, we have a web page, a Facebook page, and contact can be made by email. So all that remains for me to say is to wish you a very happy and virus-free Christmas.
Thank you very much, Alan. That was so interesting. Yeah, I love a bit of thematic science to end off my year. So what I suggest we do, the time is currently 22 minutes past 8. Shall we take a quick break to maybe pop to the loo or get a cuppa, and then we'll meet back again at past. So in 8 minutes time to do some Q&A, if that sounds good to everyone.
Brilliant.
Lovely jubbly. Okay, see you again at 8:30.
Hello, Philippa.
Hello, you've got a blue hat.
Of course, mine, yeah, because it's an Everton one. I wouldn't wear red, goodness.
Well, I'm very traditional. I almost put my Christmas jumper on, but I didn't. So I thought about it.
Annoyed the dogs because they don't like the bells. or the one's asleep and the other one's found something to chew.
Can't see John.
I've lost everyone else. I'm fiddling about trying to
you
get
lost
the
everyone,
screen
John?
sharing back.
We saw you earlier.
Yeah.
I had trouble getting
Oh,
on this time.
thank you for the card, Philippa.
Oh, it's fine. It's fine.
What happens if I hit that?
Very eco-friendly rather than putting a stamp on a card.
Yeah.
John, is the space station going to be going over visible? Do you know?
If we have a clear night, probably. I'm not sure the time and date though.
No.
It should be visible almost every night, shouldn't it?
I'll have to have a look.
It doesn't change orbit, does it?
No.
Depends whether
No.
we're in daytime when it goes over or not.
Hmm.
I'm not too sure actually.
I remember a few years ago, it went over just after 6 on Christmas Eve, which was fantastic.
Well, as it's pretty regular, it should do the same again, shouldn't it?
I must check.
I'm fiddling about hitting buttons here, trying to get the screen share back, and I've not managed it yet. Have anyone any ideas?
I've got no idea.
No.
Down,
I don't know how to do anything.
down at the bottom, there should be something that says start video.
Oh, yeah. Yeah. Chat.
Yeah, where it says participants.
I've just clicked on
Have a
start
look
video here. It says cannot start video. Failed to start the video camera. Please select another video camera in settings. Oh dear.
Oh,
Settings.
that's too hard for me, don't know.
No, I don't know either.
No, I don't know.
It's all guesswork with you, I'm afraid.
You on a laptop, John, or is your camera external?
Well, I'm on a laptop, yeah. The camera's built in.
Strange.
But you came up before. We saw that you actually appeared at the beginning.
Yeah, I know. Yeah, I just don't know what's happening. I've got myself back, but I don't want that. I want a screen share system.
Try and switch to.
I've only got the one video camera, so I can't select another. That's Skype, whatever that is. Zoom.
And I want to switch to a different speaker, so I'm going to just have a see if I can get to my Zoom bit.
No problem.
Meeting controls, is that what I need?
You can stop screen sharing if you like, Alan, just for the Q&A.
Okay, so how do we do that now?
You want to pop up to the top of the screen and you'll see a red button that says stop screen share on it.
Oh, yes, I see. Yeah, oops, gone. Hold on. Stop screen share. There we go, that's better, isn't it? Right, now I want to just switch off my headphones.
I don't need those on, let's see.
For people still in the Zoom call and those of you still watching on YouTube, we'll be doing a question and answer section in a couple of minutes' time, just when Alan's got his speaker sorted out. So feel free to leave any questions in the comments section, or if you're on the Zoom call, obviously come on mic, and we'll get them sent over to Alan. Cheers.
Hello. Yeah, I think that's working. Can you hear me?
You're very quiet, Alan, but we can hear you.
Quiet, am I? This is always a problem we have with the computer. Hold on, let me just see if I can find my sound settings. Microphone. 2 apps are using your microphone, are they? So what are they?
You are audible, Alan. You might just have to speak up a bit.
Okay, I'll shout.
Lovely, jubbly.
Okay.
All right then. So thanks, Alan, for that wonderful talk. Does anyone have any questions that they want to start off with?
Yes, I have one. Alan,
Yes.
is there a limit to how many mutations a virus cell can adapt to? Any
that
given
again?
particular one?
Number of mutations. How many?
So the
Sorry, any particular virus, or are you thinking of
Any particular one, because they're all different, aren't they?
Right, it depends on the viruses. Some are fairly fixed and don't mutate, and other viruses are fairly free to mutate. So, for example, our influenza virus can mutate loads and loads of times. I mentioned the avian influenza. There's about 40 different types.
Oh, blimey.
And they all mutate.
Yeah.
So the New York one I mentioned was, I think, N7H5 or something like that, and this current one is H5N8. And if you have a look at the list of them, there's about 40 different subtypes. But other viruses stay fairly fixed in their mutations. They don't mutate readily. COVID, we don't know yet whether that mutates readily, but there's hints that it is doing so. Before the meeting started, we talked about mink, and COVID virus has mutated to infect the mink, and that mink then can spread it back to humans. So you get zoonosis and reverse zoonosis.
Yeah.
Is quite a troublesome sort of issue, we think, but from what I heard on the television and radio, the scientists said, oh, they just need to dial in the new virus's DNA, or RNA, should I say, into their vaccine production template, and they can create a vaccine, adapt the vaccine like they do with our human influenza. They can adapt that each year to modify the vaccination and what it protects us against.
Yeah. It does seem somehow an evolutionary dead end if a virus kills a host, doesn't it?
It is, and generally speaking, it's not in the best interest, as you say, for a virus to kill its host off. But on the other hand, if you go back to that example of which I mentioned, the last of the insects, the caterpillars, when they kill the host off, the host dies and spreads its virus onto the next lot of caterpillars down below. So it may not be necessarily a bad thing for viruses to kill us off. Ebola virus in humans, it's spread by the secretions that come from a person. It doesn't matter whether they're dead or alive. For a while, those viruses are still just as infected, and most of the spread occurred when people were burying their dead relatives.
Any other questions for Alan?
That's killed everybody off, hasn't it?
Yeah, I was fascinated by the bees going to the fungi for a cure. I thought that was fascinating.
Yes.
I assume there's research into that to find out what it is and whether we can use it.
Yes. Yes, this was from one of the BBC Sounds podcasts. The guy who's written a book about fungi, and this is an observation that he has made, and people didn't realize why these honeybees were visiting these wood piles, but apparently they would collect some of the chemical that the viruses were producing, or maybe the virus itself, I don't know. They might be taking the virus back to the hive, and then the virus, like some ants, collect fungi. Sorry, did I say virus? Collecting the fungi and taking the fungi back to the hive, like some ants grow fungi in their ants' nests. And maybe they do that because there's some antiviral product, but I think they eat the fungus, actually, ants do.
We could send a link
we'll try and find
to the Radio 4 programs.
Yes,
they're on the sound.
Yes, so BBC Sounds. We'll try and find you the link and send it on.
Yeah.
Put it on the
to everybody
on the website or the Facebook page.
Interesting man's work.
Yes.
And a new book.
I have a question for you, Alan, around the rabies virus, if you know much about it. So I always wondered, as far as I'm aware, there are certain species of animals that are more resistant, things like the American opossum, for example, whereas rabies is almost always fatal to us, and things like dogs. So why are some species more resistant to that virus than others?
Good question. I don't know. Bats are resistant to a heck of a lot of viruses, including they can carry Lissa virus. Yes, I think that was one theory, is that their metabolism can slow down or alter, and maybe that's not very conducive to either the virus survival, or it may not be conducive to the virus causing disease, but it's well known that if you handle bats, you handle them very carefully, because they can actually give you rabies. Yeah, and of course,
Daubenton's, isn't it, specifically in the UK?
I'm not sure which bats they are, but some people have died in Scotland from rabies picked up from bats there. It doesn't happen very often, but it's advised that people who handle bats have rabies vaccination, and also wear Kevlar gloves when they're handling the bats, so they don't bite them or get saliva on their hands. Yeah, but it's that was probably my last talk, wasn't it?
Paul Wilkinson.
Paul Wilkinson, yes.
We have a question from Simon Keeling, which is, how is bushmeat getting here? Is it individual travellers in relation to the spread of viruses through bushmeat?
That again, I don't know the answer to that. I would think that the amount that's being imported is going to either come in with a lot of people, or it's going to be officially imported and used for people who fancy that sort of meat. But it's pretty weird meat, and obviously the trade in primate bush meat would be banned in this country. It wouldn't be allowed.
It is much more of an issue in places like Africa, Southeast Asia, where the bush trade's more lucrative. Yeah.
Not required by zoo.
I guess a follow-on question that then, Alan, is if you had to come up with a sort of single policy that would reduce zoonoses and pandemics caused by viruses transferred from animals to humans, what would you do?
That's $1 million question.
Indeed.
Yeah. Well, obviously, I think mixing.
You'd self-isolate.
Yes.
Going to lockdown immediately.
The situation that occurs in China is you have a mix of different species of animals, live animals, being sold in a market, combined with the sale of killed bush meat, and because you get this mix of animals, the viruses by natural selection or may take advantage of any mutations that they have and spread from one animal to the other. So we know that these influenza, Asian flu and so on, often starts off in birds, and then it translates into pigs, and then often ducks it is in the markets. The pigs are nearby, it spreads to the pigs, and then from the pigs it's very easy for the virus to get into us, and once it's in us, then we go on our travels and we spread it around. So the other way of stopping virus pandemics is to stop travel.
Holidays at home then next year.
Yes. or hold your breath if it's an airborne disease. Right. No, I mean we're doing what we can, but I think the government is in a very difficult position in knowing how to second guess what this virus is going to do, and the best ways of trying to keep us safe is not very good for our economy. So you have to balance all these issues out. That's why vaccines are made for some animals and not for others. For example, there is a foot and mouth disease vaccine, and it was used, I think, in 2001 to what they call do a ring vaccination. So you have your hotspot of cases, which you've slaughtered out, but to stop it spreading beyond that farm, you would then vaccinate all the healthy animals around the farm or around a 3 to 10 kilometer ring all around that farm, and then you've got a fire break. The virus can't get past those vaccinated animals because they don't propagate the virus. Now, track and trace ought to have been doing something like that. We should have been able to find out the source of the infection and then isolate that source and remove all susceptible people from around that source, and that would then create its own fire break. But of course, it's not worked. And that might be because we can't test accurately, or the test.
So many people asymptomatic.
Or there's so many, yes, or there may be so many asymptomatic carriers. Again, see, we don't know. It's a new virus. And we had this situation with parvovirus. I was a new graduate when that first came out, and we were finding these dogs in kennels and so on, greyhound kennels, 10, 15 dogs out of 100 ending up dying very suddenly within 24 hours. And, we were panicking as to what to do about it. And the bottom was there. So it became, again, economically quite important. And of course, more than economics, because there were some people's pet dogs, and people don't like losing their pets. So there was a sort of a psychological, no, I don't know if that's the right word. Yes, there's an emotional value there that had to be protected. And so vaccines were the right thing to do.
So you've seen this before. I guess the real question then is, will we learn this time?
I think we do. Yes, we are learning, but we're learning as we go along. And I think we're doing a pretty good job. I mean, the vaccine manufacturing companies have really been put through their paces, and they've come up trumps. Unfortunately, there's no coronavirus vaccines elsewhere that we could have tried out. I mean, we do have cat and dog, or the Americans have a coronavirus vaccine in cats. Sorry, so just saying. Yeah.
TB. It was a little bit muted about whether BCG vaccination might protect some people. But
yes.
Just was a little snippet in a paper, and then it was gone. Never said again.
Going back to these coronavirus vaccines in cats, unfortunately, they're coronavirus group A, and they don't protect against coronavirus group B. And the interesting thing about the coronavirus vaccine is made in America for cats. We don't use it in this country because all it does is stops the symptoms of disease, but it doesn't stop the virus, which may be second best, but that could be an option. If the vaccine may only prevent disease, but it may not stop the virus. We don't know yet. So it's a case of watch this space. And of course, HIV virus, they've never been able to get a vaccine because that mutates at a rapid rate of knots. So every time they get a vaccine that works, the next week, a new one emerges.
So I guess it's a case of see what 2021 brings.
Yeah.
Well, they seem quite confident in the protection levels, the virus and vaccine manufacturers.
Fingers crossed then. And on that note about 2021, does anyone else have any questions before we bring this year of programs to a close?
No, I just want to say thank you. It was fascinating, but particularly thinking of viruses as not all being harmful, because
Yeah.
when you teach A-level biology, you talk about good and bad bacteria. You never talk about good and bad viruses. So
No.
the bit about bacteriophages and the antibiotic resistant bacteria, that's really good. And I hadn't thought about that at all. So that's
Well,
very
that may be our salvation, because one of the big worries for the medical profession is running out of antibiotics.
Yeah.
Because those antibiotics are becoming less and less effective, or the bacteria are getting more and more clever because of selection pressures to overcome these antibiotics. The Russians certainly have been using them very successfully, but you have to actually find the right bacteriophage. It takes a lot of time, but if you find the right bacteriophage, you can multiply that very readily, as you saw, 320 minutes. If you find the right one, it will do its job, and you will cure somebody with when no antibiotics are helping.
So I shall be doing some research.
Yeah, the Russians have been using them for a long time, because I remember not that long ago when I was at university, not long ago, but we learned about bacteriophages, and they were saying that they were going to be very soon this sort of helping all humanity. So hopefully it will do.
Well, the other thing on the not on the horizon, but actually in use now, is the use of adenoviruses to actually carry in genetic material.
Yeah, that's fascinating.
That's about the only one that we would use at A-level, about using them as a vector, definitely.
And those viruses actually can the body protected. The normal cells don't get affected because those viruses have got a human gene in it, which these immune cells recognize, but the cancer cells don't, because they're not built to recognize antigens. So these viruses then invade the cancer cells and kill them off.
Yeah, and using something like the flu virus to sort of take a good gene to cure something like cystic fibrosis. That's been tested as well, isn't
therapy.
it?
Yes. And this has all happened in the
So they're
last
not
20, 30 years.
One using the adenovirus in chimps or something. You know, I've read so many things and I'm getting all mixed up now. But something about, because they didn't want to use the coronavirus live, but they could use the adenovirus.
I think it's adenovirus they like to use at the moment.
One of the vaccine producers.
Sounds like we've got a brighter future in store. And that's the message we can take.
We hope so. And what does tend to, I mean, if you think of other major pandemics in the past, they have tended to die out in the end.
It just takes time.
But it requires good herd immunity, and you can only allow herd immunity if the virus isn't too pathogenic or virulent. If it's pathogenic or virulent, then you have to do what the government's doing at the moment and try and protect us while we find some answers to work in a different way. I mean, my attitude initially was, well, let the young people get it because they don't get affected by it, build up the immunity in them, and then the virus will die out in these younger to middle-aged groups.
Sorry, Ben.
And then we can expose them to the granny and granddad, you know.
So when I'm having a lovely week in bed with no taste or smell, I
but
can
it's not
thank you.
out that way, no.
Can say it's all right. Alan says it's going to be fine.
Yes, don't quote me. Yes.
All right then, everyone. On that cheerful note, thank you so much, Alan, for a really interesting session. It was a really good one to end the year.
Been a pleasure.
Thank you.
Thank you to everyone on YouTube that's tuned in this evening. So the next meeting from now, which of course in this age of viruses will be of course online again going into next year, but who knows, maybe by the end of next year we'll be all sitting in person with each other again. We'll have to see, won't we? But we're going to have next time, January the 17th, I think it is, the third Thursday of January, we've got Will Townsend, who is a young pioneering physicist who is going to come and talk to us about some of the things in the world of physics that interest him most. It's sure to be an interesting one, but until then, from everyone at the Womborne Scientific Society, have an absolutely wonderful Christmas. Stay safe, everyone. Enjoy it as best you can, and we shall see you all in the new year, 2021, the year of virus curing.
Yes.
It's
And
the
you, Ben?
21st. It's the
21st,
21st.
Ben.
January the 21st, my apologies.
21st, yeah.
It's a good thing that, people that know more than this are here. All right, everyone.
It was decided on the committee meeting, didn't it?
Okay, so
Right.
have a wonderful holiday, and we shall see you all next time. Take care.
Thank you.
All right.
Thank you, Alan. Okay.
Thank you.
Thank you. Bye.
Bye.
Thank you, Alan. Bye. Bye, John. Bye.
Bye. Bye, Ben. Bye, John. Bye,
Bye.
Philippa.
Bye.